The C4 pathway begins in the mesophyll cells. Carbon fixation of a 3-carbon phosphoenol pyruvate (PEP) molecule with a CO2 molecule takes place. An enzyme called PEP carboxylase catalyzes this reaction, forming a 4-carbon molecule called oxaloacetate. Oxaloacetate gets rearranged into malate. The malate product is transported through plasmodesmata from the mesophyll cells into the bundle sheath cells. Here malate is broken down into pyruvate and CO2. This allows CO2 to accumulate in the bundle sheath cells. Rubisco picks up CO2 and incorporates it into the Calvin cycle. Pyruvate is rearranged into PEP back in the mesophyll cells. In C4 plants, carbon fixation occurs in the mesophyll cells, and the Calvin cycle occurs in the bundle sheath cells. Under hot and dry conditions, Rubisco has a higher affinity for oxygen than carbon dioxide. The ability of PEP to use CO2 under hot and dry conditions makes the process of photosynthesis more efficient. The adaptation of the C4 pathway maintains a high concentration of carbon dioxide molecules, so that carbohydrate production levels do not decrease. This prevents the plant from entering photorespiration and makes it suitable for living under these conditions. CAM plants are generally found in regions where the conditions are very hot and dry, such as deserts. They are known as succulents because they store water. CAM stands for crassulacean acid metabolism. CAM plants evolved to close their stomata during the day and open their stomata at night. Like the C3 plants, carbon fixation and the Calvin cycle occur in the mesophyll cells. But in CAM plants, these two processes take place at different times. This is known as the CAM pathway. During the night, CAM plants take up CO2 and, through carbon fixation, generate a range of organic acid molecules that get stored in vacuoles until the stomata close. During the day, the light reactions produce the ATP and NADPH, providing the energy to fuel the Calvin cycle. The carboxylic acids release CO2, allowing the Calvin cycle to produce carbohydrate molecules.